All right, so what does the fusion pore look like? These are even fancier EMs than Heuser and Rees did. This is a much more contemporary one done with some fancy shadowing techniques and very small or very high magnification. You can see the presynaptic membrane and the vesicular membrane and a postsynaptic membrane in this EM. And the interpretation of these electron microscopists is that one can begin to see a dimpling in the plasma membrane where the vesicle is beginning to fuse, and maybe a little volcano-like or plug in the middle of a hole structure here. And to confirm this idea, they took isolated synaptosomes, which contain pinched off presynaptic nerve terminals, and they fixed them onto a slide and then looked at them with an atomic force microscope. Now, atomic force microscopy, instead of using optics, uses a little bouncing piece of metal that's nanometers in diameter and length, and the little bouncing piece of metal will bounce over a biological surface, and based on the amplitude of the bounces that can be sensed by a very sensitive needle that is holding this very fine little tip, one can reconstruct high points and low points, peaks, offs, shadows, etc., and basically by bouncing very carefully over the entire surface, systematically back and forth, reconstruct a surface structure. And this is a reconstruction looking into a fusion pore, where they find this very plug surrounded by 8 different bumps and maybe some little ridgy things to hold this plug in place. But it's an interesting view, and I'm not sure that we know exactly what the constituents are at this point in time, but it's a really fun view. All right, so what about some of these reconstitution experiments? This technique I'm calling FRET. That's fluorescence, resonance, energy transfer. All right, so the reconstitution is because we're putting the known proteins, the vesicular snares, which is synaptobrevin, and the target snares, which is SNAP25 and syntaxin, into separate liposomes of known lipid composition. And in this assay, we've also, the experimenters have added fluorophores to the synaptobrevin containing vesicle. And they've added 2 fluorophores, not one. A green one called NBD and a red one called rhodamine. And the idea here is that when the fluorophores are very tightly packed, because these liposomes are probably 100 nanometers max, maybe less, 20, 40, 50 nanometers, the size of synaptic vesicles. So if you get a lot of fluorophores in there, they're very tightly packed next to each other. There's no elbow room. And when you stimulate the green fluorophore with 488 nanometer green light, and you would expect it to emit at slightly higher wavelengths, and that emission light immediately is matched to be the stimulatory wavelength for the red dye. So the emission from the green dye is immediately absorbed by the red dye molecule, and it then emits at a higher wavelength, so that one will see very little green, because that green emission is immediately absorbed and transferred to the red molecule. So we initially have a very low green fluorescence. In the absence of calcium, these two populations of liposomes, when mixed together, don't really fuse. And if one adds calcium, they fuse a little bit, but they really need the combination of calcium and that C2a and C2b domain from synaptotagmin. So if one adds that fragment of the synaptotagmin molecule, you don't even have to give it the transmembrane domain, but just the C2a and C2b domains, and mix those in with the two populations of liposomes. Under those conditions, in the presence of calcium, one can get fusion. Now, since the synantaxin liposomes didn't have fluorophores in them, when the two different kinds of liposomes fuse together, you get bigger liposomes, and those bigger liposomes, the red and the green molecules, are farther apart. So the emission from stimulating the green fluorophore doesn't get to stimulate the red fluorophore because it's not next door anymore. And instead, it just becomes green light. So we see much greater green fluorescence, and we see very little red fluorescence in a population of fused vesicles. So what does this look like experimentally? Here, the y-axis is the donor fluorescence, which would be the green fluorescence. So in the absence of calcium, and in the presence of the calcium chelator, EGTA, and in the presence of synaptotagmin, very little increase in green fluorescence, very little fusion of the two types of liposomes. However, when one adds both calcium and the synaptotagmin fragments, then there is indeed an increase of donor fluorescence. Now, at the very end, if you add something to absolutely lyse the liposomes, then you can get the maximum amount of fluorescence out, and you can see the maximum fluorescence step here at the very end. And if you know the diameters of the vesicles and what the concentration exactly is of fluorophore, you can actually count how many times vesicles fuse to get the increased fluorescent curves that you've got. But notice what the time scale is here. The problem with this kind of a reconstitution assay is that it's taking place over the course of a couple of hours. We think of synaptic vesicle fusion as something that can happen in several milliseconds. So this is problematic. Is this really going to be a system that could work at a nerve terminal to give us the kind of rapid neurotransmitter release that we need to keep information transfer in a neuronal circuit happening? Not such an easy question. So fast forward another 7 years or so, and let's look at some other proteins that are known to interact with the snare pins. On the left is of our familiar vesicular snare, synaptobrevin, or plasma membrane target snare, syntaxin in yellow with the NSF, or the SNAP25, excuse me, folded and bound to the syntaxin, not shown with the twist. Usually the diagrams don't show the twist. But it is energetically favorable for the synaptobrevin to begin to bind and to twist. And then there's another molecule that gets into the act and twists up, and that's called complexin. And complexin for a long time was very hard to understand because sometimes it prevented fusion and sometimes it facilitated fusion. And so there was quite a bit of controversy, and people argued about complexin up until a year and a half, 2 years ago, quite a bit. It turns out that complexin normally only half binds. And these are all alpha helices here. So all the alpha helices are kind of twisting together. But complexin only half zippers, because we think of this process as starting at the N terminals and zippering up towards the C terminals, or towards the direction that would bring the vesicle, the synaptic vesicle, very close to the plasma membrane. So it's only under condition that complexin can fully zipper that we actually begin to see fusion occur. And under those conditions, then the working hypothesis is that the synaptobrevin and the syntaxin unbend, and these what had been bent portions undergo a conformational change and become alpha helices, so that helps to maintain the fusion pore open, and we begin to see transmitter come out. Now let's take a little larger look here at the complexin, which is now the cyan or light blue molecule. This is just a diagram of where we were before, and I'll explain the little bits of differences. The C-terminal domain of complexin is an alpha helix, and that binds there, as well as the central helix. But there's this accessory helix, and that portion is not normally bound to a snare pin on the same snare pin as the central helix in the C-terminal domain. So complexin is actually binding in an anti-parallel sense in terms of N to C or C to N to the other molecules. And this unstraightening out of the snares and the fusion of the membrane, which happens only in the presence of calcium, you can see added to this diagram are the C2A and the C2B domains of synaptotagmin, whose tail into the and anchoring into the synaptic vesicle membrane is not depicted here at all. But somehow or another, the energy released when the calcium binds to C2A and C2B helps to move this process forward. Now, the other thing I didn't say about C2A and C2B is that when calcium is bound, and then the C2A and C2B domain also has phospholipid binding properties. So when calcium is there, and we know that calcium is very attracted to the unstirred layer of lipids, because the lipid head groups have negative charges, so the high density of positive charge on the calcium ion attracts it to that unstirred layer right over a membrane. When you get 2 to 5 calciums bound in the C2A and C2B domains, 3 each, these head groups bury themselves in the plasma membrane, again causing strain on the curves in the lipids, giving them an energetic reason, perhaps, to change their curvature. And rearranges itself in such a way that it could indeed, with a number of snare pins arranged around the bottom of a synaptic vesicle, actually distort the membrane enough and have enough force to cause that fusion pore to open. And this is basically supposed to be the opening of the pore that you're seeing in the right-hand diagram. So it's a lot of conformational change consequent to the calcium binding to CA2 and C2A and C2B on synaptic. So the idea is that complexin would be acting as a clamp, but then also a switch. Now the problem there is there's another protein that's been shown to act as a, quote, clamp and prevent synaptic vesicle fusion, and that's MUNC18. So how does MUNC18 fit into this story? Monk 18 binds to the entire set of snare pins again, but not really. It really binds to the very far end terminal of syntaxin. So syntaxin's involved in the snare pin, and actually here on the right you can see the amount of twisting. It's not all that much between the different proteins. But syntaxin is much longer than the part that's bound, and the end terminal, which has some internal self-binding regions, does indeed bind to Monk 18. And I have seen diagrams where Monk 18 is in lots of different positions. I don't think anybody really knows where Monk 18 binds. The picture in the Candell textbook shows it kind of riding the snare pins like you would ride a horse and holding them down against the presynaptic nerve terminal membrane. This particular picture I chose because it shows Monk 18 kind of capping the end and maybe keeping the twisties of the snare pins from untwisting. I don't know that it has that function, but at least this picture made sense to me. But in reality, I don't know that we know where Monk 18 really is. The complexin here is the two-tone pink in this diagram, but one can think about there being a need perhaps for multiple proteins to act as regulators of release and to prevent release, especially if release is this downhill, energetically favorable process of conformational change, which does not require expenditure of ATP in order to cause vesicular release. If the energy is just spent getting the machinery in place, and then calcium triggers this very fast exocytosis, this transition in the lipids and proteins to open a pore membrane, or the double membrane, and make a pore, then you don't want that to happen all that often. You only want it to happen when those calcium channels open, after the action potential has gotten to the nerve terminal. So there may be roles in regulation and prevention for Monk 18 and complexin, and maybe even more proteins. There's some Monk 13, and there's a lot more on the list. All right, so what if energetically everything's favorable to twist up all of the snare pin proteins and cause the pore to open? How do we rescue this vesicle? Or how do we get it ready again? And how do we get all of these expensive proteins, which are involved in the snare pins, untangled so the vesicle can either fuse with the plasma membrane or do whatever its recycling thing is going to be? And that, turns out, requires energy. The untwisting of this process occurs via some other molecules we haven't talked about. NSF is the name of one, and SNAPs are another family. And this SNAP, the SNAP doesn't stand for the same thing as in SNAP 25, so it's an unrelated SNAP. Sorry. And this does utilize ATP in order to untwist the snare pin complex and reset at least the synaptobrevin ready to be used again. The other interesting piece that I want to call to your attention in this diagram is that in terms of assembling the syntaxin, Monk 18, and SNAP 25, a whole number of chaperone proteins might necessarily be involved. This is cysteine string protein. I don't remember what SGT stands for, but heat shock protein 70, are all involved as chaperones in this process. And another very interesting protein, alpha-synuclein, appears to be bound to the vesicle and have some interaction with synaptobrevin. And while we don't know the exact molecular interactions of these proteins, we certainly know that abnormal expression levels of alpha-synuclein and abnormal expression levels of cysteine string protein, both of which lead to neurodegenerative diseases and cognitive dysfunction. So very interesting loose associations here that I just wanted to point out. All right, so this is where I'm going to stop on the story of synaptic vesicle exocytosis, and we'll pick up in the next video with the recycling stories.